Hello and welcome to this video tutorial of the linked data roadmap of the Cancer Genome Atlas. This roadmap uses RDF and Sparkle to index the biomedical big data resources available in the TCGA's open access HTTP repository. Let's begin by going to the source code repository for this roadmap. This repository is at https github.com/tcga/roadmap. Here you can see the source code files used to generate the roadmap, which are freely available, as well as a README file, which has a description of the schema, instructions for installation, and the license. Now at the top of the roadmap, at the top of the README file, we have a link to the raw data source that was indexed. This is the TCGA's open access HTTP repository. You may navigate it by clicking on one of these disease studies here, clicking on the center type, the center domain, the platform, the data type. You see a list of archives containing that type of data. Click on one of those and you will see the data files for that particular data type. If I click on one of these, I now can see the actual raw data. Now, it's significant that we're doing this in a browser because this means that this data is now available to the browser for use in applications, as we might see a little bit later. However, it's a little bit unwieldy to have to navigate it by going through this technique, and that is why we created this linked data roadmap for all of the data files that are available in this open access repository. Here we have a class diagram highlighting the schema that we use to represent the files. A file, a data file, is the core entity that we sought to represent, and for each data file we captured the data type, the center domain, which is the center that it came from, indicated by its domain name, the archive, the platform, the disease study, and the center type, which type of center the particular center is that this file came from. In addition to this metadata, we also capture the name as an RDFS label. We capture three dates: the date last modified, the date last seen, and the date first seen, as well as the URL of the file in question. These last five properties are also captured for each directory that represents a data type, a center domain, an archive, platform, disease study, or center type. All right, let's take a look at the Sparkle endpoint. This Sparkle endpoint is available at uab.mathbiol.org/TCGA Sparkle. That will take us to the Allegrograph instance that we currently use to house this roadmap. So let's begin by running just a brief query to see what types of data are available in this repository. So I'm going to say select distinct type where TCGA, I'll just say wildcard a type. A in this case is shorthand for RDF type. Let's execute this query. So here we can see that we have archives, files, platforms, data types, disease studies. Here we see a second instance of the word file, only this one is lowercase. Here we have archive uppercase and archive lowercase. This is due to some slight modifications that were made to the schema. However, in order for backwards compatibility for applications that were already making use of this data, we kept both, and we enabled reasoning to ensure that, we used reasoning to ensure this backward compatibility. So let's take a look at some example queries by clicking on this overview link. When we go to the sort of repository homepage that Allegrograph creates here, we can see a number of predefined queries. So let's select the top one here, platforms by disease. The goal in this query was to correlate the list of platforms with the list of disease to discover which diseases are analyzed by which platforms and vice versa. So if we click on this link, we'll be taken to that query. You can see here that it also is available as a URL. So here we're saying select distinct platform and disease. That means distinct combinations of platform and disease, those variables. And in order to get the name of the disease, we are selecting for a given file. Remember, everything is centered on files here. For a given file, we are discovering all of the platform IDs that file has related to. And for each of those platform IDs, we are getting the name of that platform as indicated by the RDFS label property. Again, for each file, we are discovering which disease studies by their IDs that file came from. And then for each of those disease IDs, we are getting the name indicated here by the RDFS label property. And just for fun, we're ordering those by disease. We could also order them by platform if we wished. Here we see the results of that query, and we can see that for the breast cancer, for instance here, we can see the list of platforms that have data available for that particular disease. If we chose instead to order these by platform, then we would see which diseases have available data for a given type of platform. In this case, RNA sequencing only has 2 diseases with data available, whereas let's load some more data here. The genome-wide SNP data is available for quite a number of diseases here, as we can see. Okay, let's go back and take a look at another example query. In this query, we're going to count the number of files available by date, or on each date, we will count the number of files that were made available on that date. Again, ensuring that we have reasoning enabled to ensure backwards compatibility with previous versions of this repository. We can see that each data file was made available, or each data file, or excuse me, each date and the number of files that were made available on that date, that were uploaded and made available on that date. Using this query here. So here we're saying select the date as well as the number of files, and we get that number of files by counting how many individual file and instances there are. And all we're saying here is select each file as well as the last modified date for that file. I want to group my results by date, and I want to order them by date descending so that I have the most recent date here at the top. And these are my data results. Now I could download these as comma-separated values or as any other type of data if I wanted to use them in an exterior or an external analysis tool. Now, just to demonstrate some potential uses of this data, we've created a roadmap available at uab.mathbiol.org/TCGA roadmap. And here at the roadmap, we have links back to the source code as well as to that Sparkle endpoint that we just visited. In addition to that, we have visualizations created of those 2 queries that we just looked at. Here's the total number of files by date. In this case, we're actually totaling the files by date rather than, totaling the files cumulatively rather than by individual dates. So here we can see the rapid growth of the TCGA, and then over the past almost 2.5 years, it has doubled in size, nearly, about every 7 months. And if we mouse over, we can see the number of files on any given date. Again, with a link back to the query that generated this particular result. Here's that query. We also have below here a visualization of those connections between diseases and platforms in the TCGA. We have the diseases listed here on the left, the platforms listed on the right. When I mouse over a disease, I can see each of the platforms that have data available for that disease. When I mouse over a platform, I can see each of the diseases that have data available for that particular platform. So for instance, if I was interested in protein expression across a variety of cancer types, I would simply mouse over the MDA RPPA core platform here, and I would see each of the disease studies that has data from that platform available. Well, thank you for watching this video tutorial of the Cancer Genome Atlas open access repository roadmap that we've developed.